and he's been doing really fascinating work on microbial dynamics. Thanks so much, Supratim and Sagar. I mean, I must say, apart from all the lot of work that they've evidently been doing, organizing everything here, you've also been really giving really kind introductions and digging in. So you've added to your work of organizing. You've also been doing all this work of digging into people and figuring out what they did. But I really appreciate it. Thanks so much. So I mean, so this is going to be a much shorter talk. So instead of going deep into things, it'll be a little quick sort of survey through a particular set of questions and problems I've been particularly interested in. But the purpose is to hopefully give you a sense of how, some of the tools that you've been seeing, dynamical systems, game theory, can be used to answer specific questions about a particular biological system, right? And what I've chosen to talk about is in a context where the tools are going to be really, much simpler than even what you've been hearing now. Chaitanya has been talking about these games with multiple players and all. I'll talk about really simple games, 2-player games, very simple stuff. So, but even I want to show you that even with, the simplest versions of these tools, sometimes in a particular biological context, you can still get some interesting insights and at least raise some interesting questions. Okay, so the particular context that I want to talk about, the particular kind of biological system, many of you might be already familiar with them, but for those who aren't, I'll tell you about it anyway, are these viruses that infect bacteria, okay? I mean, these are actually really important organisms because as you know, bacteria, really set a lot of the dynamics of what happens in this world, and these are major predators of these kinds of bacteria. And people have been studying bacteriophage, as they are called, for decades. Bacteriophage, as some of you may know, were the foundational organisms which led to a lot of modern molecular biology and our understanding of how genes work and regulation of genes work. So what I'm going to be talking about are these viruses which infect bacteria. And for those who haven't seen, images like this, it's kind of fun for you to look at them. They really look like sci-fi stuff. This is an example of an E. coli bacterium which has been attacked by one of these viruses, which, this is a lab example where it's been attacked by a lot of viruses, but even a single virus of this kind can attack a single bacterium and create a scenario like this. What's happened is this virus has sent in a DNA, its own DNA, injected it into the bacterium, and taken over, hijacked the whole machinery of the cell, hijacked it to make many copies of itself. And this poor cell is eventually going to burst, and all these, hundreds of copies are going to come out, and there'll be new viruses which will go on and infect other cells around, okay? So you can already see this is a kind of a predator-prey system, but it's a little bit of an unusual predator-prey system, unlike, say, the classic ones we know, because imagine if a fox eats a rabbit, and eating a single rabbit, it produces, hundreds of new foxes, okay? That's what's happening with these viruses and bacteria. A single virus can infect a single bacterium and produce hundreds of offspring. So, many questions already arise. You'd start wondering, how is this even coexisting? Why aren't the foxes killing all the rabbits? What's going on? And these are valid questions. One of the strategies, there are many questions around this coexistence question, but one of the strategies that is used for viruses and bacteria to coexist is that some viruses don't necessarily always do this virulent strategy of going in, taking over, making hundreds of offspring and killing the cell. Sometimes they can go dormant. So there's a whole class of viruses called temperate bacteriophage, which when they insert their DNA, by the way, just as an aside, I should say there are many kinds of viruses. You would have heard about them even in a human context. There are viruses that contain DNA, there are viruses that are RNA viruses. We won't, in this talk, have time to go into all this. So just imagine, we're talking about viruses which have a DNA string inside them, which encodes viral DNA, viral proteins, and so on. The bacterium has its own DNA. The viral DNA is injected inside, and now a decision happens, okay? A decision between two pathways. One is what I was showing you earlier. The viral DNA starts producing viral proteins, which take over the machinery of the bacterial cell in such a way that they start replicating the DNA of the virus. They start producing all the proteins and stuff required to produce new viruses. And eventually they burst the cell and hundreds of offspring come out typically. The other thing that can happen is they can go dormant. They don't do all this stuff. They sort of sit quietly in a way. And in fact, in this kind of example, they in fact insert their DNA into the DNA of the bacterium. And subsequently, the bacterium, this is pretty stable. This is a pretty stable state. It lives more or less happily ever after unless there are specific triggers that cause this to come out and jump into that mode. But as the bacterium grows and divides, its genome duplicates, and the genome of the virus duplicates along with it. And as I said, there are specific triggers which can sometimes make this jump out and jump there. But this is pretty stable. So a decision is made here whether to go this way or this way. All right? Yeah. Of course, there are some situations where what's called an abortive infection happens, and neither of these happens. Something happens along the way, and there are also, of course, and I'm not going to talk about this now. There are mechanisms that the bacterium, of course, has evolved. I mean, this is a evolutionary arms race, right? The bacterium has defenses to detect the intrusion of foreign DNA and deal with it, try and destroy it, and these can be either active or passive systems, but in that case, nothing will happen. But let's say an infection proceeds and the viral DNA is not destroyed. For these kind of phage, these two things can happen. All right? And I've been really interested in this system for a long time because it's a really almost a minimal example of a biological system that makes a binary decision. All right? And there are many wonderful questions you can ask about this at many scales. I mean, don't worry about this very dense type, but the idea is to show you that you can think about questions, you can ask questions about this at a subcellular scale, at the scale of what are the genes and networks that are encoded in the viral genome. How are these viral genes regulated? What feedback loops are there? Why is it? How does that even allow you to have these two states, okay, that you can go into? At the cellular level, you can ask more sort of optimization questions. What is the effective strategies that this leads to at a cell level? What is the cell trying to do? Is it trying to optimize something? This is often where, and this is where the rest of the talk will sort of focus, where you can bring in tools from game theory, and you'll see in a second what I mean. But you can also ask at the population level, and that sort of also ties in a little bit with what Chaitanya was talking about. You can think about, of course, these games are not happening inside a single cell. They're usually, it's a whole population of bacteria that's attacked by a population of viruses. And you can ask, how does this kind of decision making, which is implemented at a molecular level, results in some strategies, effective strategies at a cellular level, how does it play out at a population level? What kind of spatiotemporal patterns do you get? How does it affect coexistence and long-term things? And of course, all of these questions are interconnected. But just to give you an idea that this kind of system is really rich. A simple decision, but it has a lot of consequences all across the board. So just to give you a little sense before I dive into this scale, where I will talk about some game theory and some kind of game theoretic analysis of this system, I want to give you a small, quick introduction to what we know at this scale. Okay, just so you know underneath the hood, what is the nuts and bolts of this. So, a very well-known example of this kind of a bacteriophage is the lambda phage. I don't know if many of you have heard it, but lambda phage was, again, like I was saying, one of these really important organisms for the development of molecular biology. It was studied a lot at the genetic level. And for lambda phage, people feel like they have a nice picture of why these two states, these two pathways are possible at all. Okay? They think that underlying this kind of system in lambda phage is a simple bistable switch where there are two proteins. They happen to be called C1 and CRO in lambda, it doesn't matter. But there are two proteins, each of which inhibit the transcription of the other protein, the production of the other protein. And this, you can quite easily see, has two, just even in a hand-waving way, you can see it can sustain two stable states. Imagine C1 is high in concentration. Somehow you produced a lot of C1, but not enough CRO. Lots of C1 will suppress further production of CRO, and CRO will remain at a low level, and this inhibition will not happen. And therefore, you'll have high C1 and low CRO. That's one state. And the other state is the exact opposite. And people have done experiments with lambda phage, and they indeed see that C1 is high in this, and CRO is low in this, and you can delve into the particular details of why this works. And C1 happens to be a protein that suppresses all the genes in the viral genome that do this replication and production of proteins that is needed to assemble more viruses and so on. But, of course, we have now been studying lambda phage for like 50 years. So we know the entire genome. People have looked at every single gene, they've mapped out what they are, and they know most of the functions of almost everything. And this picture has become more complicated. This is a picture of what we know about lambda. Now, I was talking about these two interactions, where C1 represses CRO and CRO represses C1, but there's lots of other interactions, as you can see. And the point of this is to show you that these simple little network pictures sometimes are not the whole thing. But a lot of what's going on is that this decision is also not just a simple decision of 50-50 stochastic decision between going this way or that way. You can imagine for a system like this that it also matters that the decision is tunable depending on the kinds of conditions you find yourself in. There must be conditions where it's better to go virulent, and there must be conditions where it's better to go dormant, and a virus that can adapt its decision depending on the surroundings it finds itself in will probably do better than a virus that is just doing a random decision, right? So a lot of this complexity, people believe, is because the virus is trying to bring in information from outside or from the bacterium and bias this decision. And so for instance, how well is the bacterium doing? Has it got lots of food, etc.? What's the temperature like, and so on and so forth, okay? So this is just to give you a little background, and keep this in mind as I progress with the story in this talk. So let's come back to this, okay? I want to sort of focus at the cellular level, and one question has been, so I was talking about, how would you tune this decision? So of course, one experiment that has been done is to try and put a population of viruses together with a population of bacteria that they infect, and just ask how many go this way, how many go that way, all right? And it's remarkable, one remarkable fact is that for a very large class of tempophage, not just lambda phage, but lots of, now this is known for lots of phage, if you take more or less a 1:1 ratio, and you put them in conditions where the bacteria are growing very well, the percentage that go lysogenic seems to be pretty narrowly confined. It's somewhere between 5 and 15%. You rarely see, 80% lysogeny or, 1% lysogeny. You see about 10%. So where does this number come from? There are multiple kinds of explanations that have been sort of put forth, but one obvious kind of explanation, which is certainly partly true, I'm sure, is that, what is this decision for in the first place? It's for hedging your bets against what kind of environment you would find. There are environments in which, phage viruses that are outside the bacteria are very susceptible to something. It could be, some kind of dangerous chemical or something that kills the viruses that are outside the bacteria. But will leave viruses that are inside bacteria, that are dormant inside bacteria, safe. But there'll be other kinds of conditions which will be dangerous for the bacteria. Stuff like UV damage, which is destroying bacteria, or some other kinds of antibiotics which are destroying bacteria. In those kinds of conditions, as a virus, you might not want to be stuck inside a bacterium which is dying. You want to be outside. So people have proposed that, in an uncertain environment, you would hedge your bets, and then this percentage of lysogeny would be reflective of how much time you see this kind of environment or that kind of environment, right? Which is fine. But we were interested in a kind of a slightly different kind of explanation, and we wanted to explore what happens if it's nothing to do with the environment, but it's to do with competition with other kinds of viruses. So let's say you have two very similar viruses that are infecting the same bacterial population, two types of, two variants of lambda phage, which are identical in all respects, and they're attacking the same population of E. coli, but they're just slightly different in the way they make their decision. One, say, goes 10% lysogenic, one goes 20%. Let's just keep it really simple, okay? What would happen? So the way to sort of study that, at least theoretically, is rather straightforward. You build a little sort of population model, which is based, in this case, these equations are really just like a Lotka-Volterra equation. You have terms for bacterial growth, you have terms for bacterial infection by the virus, and then bacterial infected bacteria. So this represents bacteria that are non-infected. This represents bacteria that are infected, which can basically make a decision between either going lysogenic, which will lead to lysogenic bacteria that can further grow and divide, or it leads to new bursts of phage that come in. So, and this will all be characterized by parameters such like, I'm not going to go into the details because I just want to give you a flavor, but you can see how I can build a little population dynamics model, right? And there'll be parameters like the growth rate of the bacteria, the rate at which they get infected, the burst size, which is how many offspring come out if the virus goes in and decides to go lysogenic, things like that. So all these parameters are there. There's a little dynamical model like this, and you can run it. So let me take you through. Here's an example of a run, but it's a single run, but it's pretty typical of what you see. The parameters that we've chosen are parameters that would be sensible for lambda phage infecting E. coli, with the slight difference that the only thing that we vary is that the chance that you will go lysogenic, okay? So there are two types of virus, two types of lambda phage infecting a single population of E. coli. We start with a very small number of E. coli and an even smaller number of both phage. We're going to keep all parameters identical because we just want to get in and see what would be the effect of having a slightly different decision. We don't want all the other things to complicate the issue. Just if all you could vary was your decision and how often you went lysogenic or lytic, what would you do, okay? So let's imagine one of the viruses goes 10% and one goes 20% in this case. So what happens? Initially, you have just a very small number of healthy, uninfected bacteria. They start to grow exponentially because there's not much phage over here, very, very low amounts of phage, so there's not much happening. As they grow, there are a few infections, and slowly, infection starts picking up, and some of these infections will produce more viruses and so on, and the virus population will grow. And that's this dotted line, which sort of eventually picks up. But eventually, as you can see, for a long time, the dotted line is so low that it makes almost no difference. The bacteria are not even seeing the phage, they're just growing nicely exponentially. But eventually, the virus population grows high enough that it starts to have an effect on the bacteria. The bacteria start to crash. What's shown here is that the number of infected bacteria, uninfected bacteria crash quite quickly. That causes a dip in the total number of bacteria. But what starts growing is the number of lysogens. I forgot to mention to you earlier that one really important property of these lysogenic bacteria, the ones with the dormant lambda inside it, is that they are immune to further infection. So you can imagine why from the network picture I gave you. There is a protein sitting there which is kind of suppressing everything, right? And keeping it in this dormant state. A new infection happens, a new viral DNA comes in. This protein is still around. It'll go and bind to that DNA also in the right places and suppress all those genes. So it's not unreasonable to imagine, and that is what happens, that any further infections just make no difference. It stays dormant. So eventually, these dormant bacteria populations start rising. The healthy bacteria have gone. If there's nothing uninfected left to infect anymore, eventually the viral populations will start crashing, right? So what happens in the end? There are no viruses left anymore. The total bacterial population reaches carrying capacity, but it's made of two subsets, lysogens of one type and lysogens of the other, and they're in some proportion, all right? So one of them win. In this case, it happened to be the 10% beat 20%, okay? And now this is where you can start thinking a little bit game theoretically. So I'm going to start speeding up a little because I don't want to go too far over time. But you can now start to think of this as a little game, okay? So who are the players of the game? It's a two-player game in this case. There are two types of viruses. So there are two players. What's the strategy space? The strategies space that each player can choose from is basically 0 to 1, the percentage that will go lysogenic, okay? That's it, very simple game. It's a zero-sum game. It's a two-player, symmetric, zero-sum game. It's like the simplest you can come up with, okay? So just to reinforce the point, you can build a payoff matrix, right? You can imagine a payoff matrix. As all of you know, you pick a strategy for phage type 2 and another strategy for phage type 1. You run this game. You start this dynamical system with these two types of phage. You run it till the end, and you can get a payoff from what happens at the end by seeing who won. And you can even quantify it by saying it's L1 minus L2, and you can normalize it in a way that payoffs are between -1 and +1 and so on. But essentially, you can give a payoff that's the payoff for phage type 1 will be higher if phage type 1's final population was higher and so on. Okay? So very simple criterion of winning also in a simple game like this. And you can build a payoff matrix and get some number out here, right? If it's a number above 0, that means the phage type 1 won. If it's a number which is negative, it'll mean phage type 2 won that game. And you can keep doing this until you build the entire payoff matrix. And then it's again going to be fairly straightforward to try and find a best strategy. Now, as you've probably seen in many of these lectures before, there are multiple ways one can sort of define a best strategy. It turns out in this kind of game, it's so simple that it doesn't matter. The Nash equilibrium is going to be the same as the minimax, it's going to be the same as the NESS. Everything's really simple over here. But just for completeness, we'll look at a minimax strategy where essentially, I mean, and you guys know, I think from the classes before, that the key aspect of this kind of best strategy is that any player individually on their own has no incentive to deviate. If a player 1 starts deviating from this strategy, player 2 can increase their payoff or decrease player 1's payoff and vice versa, right? And it's pretty straightforward. So let me immediately show you for, yes. No, there's just, no, this is the space of strategy. It's not that they have to sum up to 1. It's the entire possibility. You might be lysing very close to 0%, but the maximum you can close is 100%. But there is no constraint involved. You could have both of them lysing very close to 1. So the payoff matrix covers the entire space. There's no constraint on what the second player can do. Okay, so this is what it looks like. If we split this, I mean, if we do hundreds of simulations for each pair of lysogen percentage for player 1 and player 2, you essentially get a very nice, smooth, saddle-like shape. The Nash equilibrium, the minimax strategy is nicely at this saddle point. Okay, and just to build a little intuition of why this is not so surprising, you can imagine why there is an optimal in the first place. So what's happening here? Go back to this game and the nuts and bolts of this game. What would be the best thing to do for a phage? Eventually, what wins? What wins is when you dominate the lysogen population over here, right? Where are these lysogens coming from? They're actually coming from here. The game actually ends really over here, when there are no more uninfected bacteria to infect. At that point, you have a certain number of lysogens here, very small number of each type. And after that, the phage can't do anything more, right? There is nothing to infect. If you infect the lysogens, nothing will happen. There is nothing uninfected left. So at this point where the uninfected bacteria have crashed to 0, the game is actually over. What's happening here is just the growth. And we've kept the growth rates the same. So it's just the initial ratio is just amplified over here. So if you won over here, you won at the end. So what do you need to win over here? You would ideally like to have lots of the infections that happen in this time to go lysogenic, right? You'd love to have a large number go lysogenic. But because I've constrained you to just have a particular number of percentage of lysogeny, if you went almost 100% lysogenic here, then you wouldn't have very many phage around in the first place. You started off with a small number, and if you were going 100% lysogenic, you'd never build up much numbers to begin with. So on the other hand, if you were always going lytic, you'd build up a lot of numbers during this phase when you're infecting and producing offspring, But in this phase, you wouldn't go lysogenic. So there are two pulls in opposite directions. In this region, you would like to go lytic a lot and build up your numbers. And in this period of time, you'd like to crash, right? And when it's crashing, you'd like to go lysogenic. Okay, I'll try and wrap up fairly quickly. So the fact that there are two things pulling in opposite directions, not so surprising. But what is interesting is that now you can start to take this game. I just showed it to you for a particular set of parameters. What really starts to get interesting is if you start to vary these parameters. And in this case, I'm just showing you an example of varying parameters one by one. And it's remarkably robust. The final optimal solution is remarkably robustly in that 5 to 15% range. And in this case, it's a simple enough game, so I won't go into the details, but you can do this analytically, and under certain assumptions, you can actually figure out where the Nash equilibrium should be, where the minimax strategy should be. And you can see that a lot of the parameters don't make a big difference to it because they come in under a logarithm or in ways that don't really affect. And it's kind of interesting to see that this pulls and pressures bring you at this 10 to 15%, which is where we see the experiments. Okay, one final thing, and I hope you don't mind, I take just a couple of more minutes. I showed you this diagram, and I made a big deal of the fact that these decisions should be tunable. But through all this exposition about this little game theory, the decisions have not been tunable, right? I've just sort of imposed that there is one that does 10%, one that does 20%, and these players choose something. And there's no way for them to impose or use any kind of information to tune this decision. So we were interested in that. So, we wanted to look at what if you expand the strategy space to allow for tuning the decision. A very interesting case is where these viruses use the number of infections that happen simultaneously to tune the decision. I'm going to cut a long story short and tell you that there's lots of nice experimental data that shows for lambda-phage infecting E. coli. In a population, you imagine a bunch of lambda-phage infect E. coli. Some of them will be infected by one. Some might be simultaneously infected by two or even three or more, right? Just by pure chance, if you're just mixing and matching and making things infect, right? And it turns out that with lambda-phage, two simultaneous infections really push the decision towards lysogeny, whereas a single infection keeps it at a very, very high likelihood of being lytic. And this is really interesting. It's somehow the phage is using information in a curious way, just by simultaneous infections to try and understand what's happening about the environment. But from the game theory perspective, you can easily bring this into this game theory sort of thing by modifying these equations. These were the equations where this was not the case. Both players, both types of phage, just had a fixed percentage. But I can very easily change the equation in this dynamical equation of infections and decisions and so on, to modify the equations to have not just keep track of an infected bacterium, but a bacterium is infected by a single one, is it infected by two, is it infected by one of that type and one of that type, and I can have the strategy depend on that. It's a much larger space of strategies. Now it's hugely expanded, and we can no longer do these very clean analytical things, but we can explore it computationally. And it turns out we find that the best strategy is this. The best strategy, the Nash equilibrium in this more complicated scenario, is to exactly what we see in lambda-phage, is to go lytic if you have a single infection, and to go pretty much lysogenic if you have two or more infections. And just to give you a little intuition there, I had told you how it would be very nice to be able to go lytic in this region and then go lysogenic here. And it turns out that this strategy is a really neat way of switching from going lytic almost always in this initial region. And then when these numbers, the virus numbers, shoot up, then you start seeing a lot of multiple infections, and all those multiple infections jump to being lysogenic. And so suddenly you're very close to the ideal switching strategy that you would want. And it's a way for the virus to find this as close to an ideal strategy, but without actually knowing what's happening, other than because it's stuck inside a virus, inside a bacterium. So it's making its best guess of what's happening. So let me stop here and just reiterate that as you've seen, I've been just trying to present some very, very simple game theory and sort of use that to make some insights into how a decision in these viruses are made. But as you saw in even the simple one, that there are lots of assumptions, right? We make assumptions when we set up a game theoretic situation about what's the strategy space, who are the players. And even as you complicate this, there are still assumptions left over. Even in the more complicated version I told you, we're still talking about 2 players. But of course, in real life scenarios, it's not 2 players. There are multi-species, there are many species of bacteria, there are many species of viruses, there are also bacterial defenses going on. So you must keep in mind that these are just ways of actually generating more questions. You get some insight. It's nice that with these simple game theoretic tools, you can get a certain insight into why you might have this kind of simultaneous infection-based strategy and switching. You get some insight into what are the pulls and pressures pushing in a different way. You get some indication that maybe competition is also an evolutionary driver and not just the environmental fluctuations. But it's always important to keep in mind that these are just tools. And in fact, it raises almost as many questions as you may have answered. So let me stop here, and thanks very much. Sorry for going a little over time. Happy to discuss any questions over tea and so on. Yeah. Sorry, I rushed. Yeah. Yeah. So they are. This is an infinity. This is a sum over the number of simultaneous infections. So BIM refers to the number of the infected bacteria that are infected with M viruses. So in principle, that number can be infinite, okay? But what we do in practice, so essentially, you can actually even, analytically even get the set of sort of steady states, even if you allow M to go to infinity in this. It's not that difficult. But in practice, what we do for computationally studying this system is we sort of truncate at a certain M and say, beyond a certain M, let's say, large enough, like 10 or something, the strategy is the same. So the percentage of lysogeny for 10 infections or 11 or 12 or 200 or 2 million is the same, okay? At that point, you can truncate it and put all of that into one bin, and then it becomes a finite set of equations, okay? So in practice, computationally, it's easy to do that. It's also pretty reasonable. It is what we see in real life in these bacteria. When they do show this kind of multiplicity of infection-based switching, really it never even goes beyond 3. Beyond 3, it's almost like 100% lysogeny. And whether it's 5 infections or 6 infections, it doesn't really matter. But in principle, the equations are infinite set of equations, but we don't actually deal with it. Does that answer? Yeah.
When you were saying decision, do you mean that based on C1 and CR, it is basically kind of doing like a reproduction thing, or so based on the environment? I mean, is it making decision, or it's like based on the environment, that's what happens?
I mean, yeah. I mean, it's certainly not. It's not a decision of the sort that there's a nervous system that's sort of clearly not. I mean, it's how you want to define the word decision. I am using the word decision here, not so much to make an analogy with, brain decision, neural decisions or human decisions. Not to say that there is free will. It's more that it has many of the aspects of a decision. It's a really simple example. So you could easily just call it a response system. That's also fine. I mean, it's more to sort of, it's not so much to say there is free will. I'm certainly not saying they have free will. So indeed, what I am saying is they are using information from the environment. There are 2 paths you can go into, and which of the paths you go into depends on what's happening. Now, whether you would, strictly speaking, semantically call that's a philosophical discussion I'm happy to get into. But that's what I mean. I mean, it's a decision in the sense of it's a control system choosing between 2 paths, choosing in the sense that, it's built in a certain way that it can go down two paths depending on what inputs it gets. Yes.
Thanks, this was so fun. I just wanted to ask, so in calculating like the payoffs from the different lysogeny strategies, how much does it matter that you're always starting them at the same population spot with just a few of everything?
Yeah, so we did try that. One of the reasons we wanted to keep things identical between the two types of players and start them at the same population level was to try and narrow down on just how much, it's an ideal scenario, how much would only changing the decision strategy affect things in the absence of everything else. But so one scenario we tried is, of course, changing that. Instead of starting with very low numbers, start with high numbers, or start with bacteria that are already close to carrying capacity, but still keep everything identical. Keep the two players starting in the game in an identical scenario, so one doesn't have an advantage over the other. That doesn't seem to matter too much. That still keeps things pretty robust. You still get, so even if you start with much more phage, unless you start with, so many phage that you actually, right from the beginning, you've just killed off the bacterium and you hardly have that build-up phase at all. So unless you squish that really, really to a very, very narrow window, you pretty much get the same answer. But if you start making things asymmetric, and you can do this in many ways, you could make it asymmetric in population as you're talking about. You could also make it asymmetric in terms of the burst size or the infection rates. Then, of course, things get more complicated, and of course, things with more infection rates and higher burst sizes get pulled in certain directions. So there, things are a little harder to interpret, and we found it easier to interpret the scenario where, at least when you start with identical viruses, and the only difference is whether they are, one is doing a little bit slightly different percentage of lysogeny, we can see the effect of that parameter on it. Does that, I hope that answers.
So what we described was a game between two different species of bacteriophages. But then here, if you have within one population, is this 10 to 15% evolutionary stable with respect to mutants? If there are individual phages that that try to cheat by being more lytic than the rest of the population.
I mean, it's a good question. I actually don't know if, I don't think anybody has actually done an evolutionary experiment where they have kept evolving these viruses to see if this 10% or whatever remains stable. So, I don't have an experimental answer to your question. Yeah, yeah, Supreet. Yeah, please. Thank you. Fantastic. So essentially, I think it then becomes what Chaitanya was talking about. We are modeling this as a simple game, a two-player game, but it really, in that kind of scenario, it becomes a multiplayer, very quickly a multiplayer. So then the two-player scenario, the framework itself is useless in that kind of scenario. What one really needs to do is then think of it as a multiplayer evolutionary game. Right. And as you've seen in his talk, there the dynamics can be pretty complicated. Thanks. I didn't know about these. I need to.